Thanks very much. Thanks very much for the invitation. It's great to be here as always, and although with 9 hours of jet lag, I'm not particularly happy to be the first up this morning. But I'll do my best to stay awake and to keep you awake as well. So I'm going to give you a summary of where we're at with the Tara Oceans project. If you don't know what Tara Oceans is, it's a little bit of an unusual project that I'll introduce to you in the first few minutes. It's unusual in that it's very much revolves around the use of a research sailboat, a schooner, that's 110 feet, 36 meters long, that is shown here in this slide. And this is the boat that we have used during this project to go out and collect our samples. The project overall is run by Eric Karsenti, who is a cell biologist at the EMBL in Germany and also at the CNRS in France. So what we've been really focusing on in this project is what we call the invisible multitude of organisms, which are the plankton in the ocean. So the plankton are basically all the invisible life in the ocean that drifts with the currents. And although they are invisible, if you add all of them up, they're actually pretty important in terms of biomass. They represent something like 90% of the biomass in the ocean. The other 10% is what you see on Discovery Channel and so on. That's the sharks, the turtles, and so on. So these organisms are very abundant collectively in the ocean, and they perform a lot of important functions. So there's all kinds of organisms going from the very smallest viruses, bacteria, protists, microeukaryotes, and smaller metazoans that drift randomly with the currents in these plankton communities. And what these guys do, all kinds of important things. Well, they're the basis of pretty much all oceanic food webs on which all higher organisms depend, including ourselves when we eat fish. The photosynthetic plankton sequester atmospheric carbon dioxide and sequester it into the ocean for long periods of time, so much like plants do on land, removing carbon dioxide from the atmosphere. Through photosynthesis, they also generate the oxygen we breathe. Something like 1/2 of the oxygen that we breathe comes from the ocean, comes from these organisms. So that's a pretty important planetary function that these guys do. And because the ocean is deep, when these organisms die, a fraction of them will sink to the bottom of the ocean, taking the carbon with them. And so this constitutes the most powerful biological carbon pump on the planet, whereby we can remove carbon dioxide out of the atmosphere. So that's a pretty important function, pretty important process that is very important for regulation of our climate, obviously removing CO2. And we know from many studies performed by many people over the decades that the plankton both affect climate over geological timescales, and they're also very much affected by changes in climate change. So it's very important to try and understand the links between these microscopic organisms and these global scale Earth level processes such as regulation of our climate. So what we wanted to do during the Tara Oceans expedition, first of all, is really to put biology at the center of what we wanted to do, which is not always the case in oceanographic research. So what we wanted to do was put biology at the heart of the project and try and study the whole collection of planktonic organisms that are present in the oceans, which is quite a big challenge because you're going from about 5 orders of magnitude of size, going from sort of the smallest viruses up through the bacteria, through the microeukaryotes, the protists, and the small metazoans like copepods and so on, the zooplankton. So we wanted to put the biology at the heart of the project, but obviously to understand the biology, you need the physicochemical context. And so we also spent a lot of time bringing in physicochemical data, as I'll introduce you to in a few minutes. So this was basically the scientific design of the project. And we wanted to do this on a global scale because the ocean is one. in principle, every molecule can be anywhere else in the ocean at some time in the future. We wanted to study the single ecosystem on a global scale. So that's why we put together this global expedition. We performed 4 years of cruising around the ocean, sailing a lot of the time. And over these 4 years, we managed to sample in pretty much all oceanic provinces. We started in the Mediterranean, went into the Indian Ocean, down into the South Atlantic, into the Southern Ocean, into the Pacific, to Tahiti and Hawaii, and then back to San Diego, through the Panama Canal, up to New York and back home. That was a 2.5-year expedition. And then subsequent to that, we undertook another expedition around the polar circle, where we went up north and we went through the Northeast Passage above Russia, and then through the Northwest Passage above Canada over over the summer season of 2013. So this is already done, right? It's finished, and we brought back to the labs a whole bunch of samples from more than 200 sampling sites with worldwide coverage. And in total, we have about 40,000 samples, which we're using for the biological analysis of the communities, together with different kinds of data sets that help us bring in the physicochemical context as well. So basically what we try to do with all of these samples that we collected is sort of a three-tiered approach, well, in parallel. On the left side, we do a lot of high-throughput sequencing. So with the size fractionated samples that we collected, going from viruses, bacteria, protists, metazoans, we basically extract the DNA or the RNA, and we do metagenomics, metatranscriptomics, metabarcoding. And then in parallel, we do a lot of high-throughput imaging, including some off-the-shelf sort of flow cytometers and basic imaging equipment that are quite routine in oceanography, together with high-content confocal imaging to get really, really nice information about the morphology of these organisms. And then in a third approach, we bring in all this physicochemical data. So with this sort of approach, we've been busy analyzing the samples, and we were happy to be able to publish our first results in a special issue of Science last May, which you may have seen. If you didn't see it, I did bring a few reprints, so just try and grab one, either outside or up at the registration desk. So in this special issue, we published 5 papers back to back describing the first results of the project. So a few key points from these papers. One is that we defined what we call an ocean microbial reference gene catalog, coming from 68 sampling sites with pretty much worldwide coverage. These genes are mainly coming from bacteria, from prokaryotes, okay? And the catalog contains about 40 million genes, which is quite close to saturation, at least for the surface, the top 100 meters or so of the ocean. It's a pretty exhaustive collection of genes coming from prokaryotes. So this collection represents something like 1,000 times more than Craig Venter's Global Ocean Survey, which was performed about 10 years ago, 15 years ago. Okay, so it sort of sets a new standard for what can be done in microbial oceanography, I think. And in terms of just DNA content, the data set is equivalent to 135 fully sequenced human genomes. So there's a lot of unknown genes in there, a lot of interesting things you can pull out. Go dig out your favorite genes and learn more about them in this data set. In another paper, we described the viral communities that we were able to fish out of a range of different sampling sites. And from this study, we basically described about 5,500 viral communities, only 39 of which were previously known. This gives you an idea of our ignorance of microbes in the ocean, in particular viruses. I'm not going to dwell on this further because Jen Brum, one of the lead authors on this study, is here and will be presenting this work in more detail later on during the week. So that's the viruses, and then concerning the protists, the eukaryotic community, we basically described these communities using a meta-barcoding approach, and we found here saturation at about 130,000 taxa using our method of OTU binning, about 130,000. So again, that's pretty much saturated. That represents pretty much everything that is there, at least in the top 100 meters or so of the global ocean. So this is more than 10 times higher than the number of formally described marine eukaryotic plankton. So 90% of these are new, and about a third of them cannot even be assigned to any known taxonomic group. So we cannot even put them on the eukaryotic tree of life. So there's probably a lot of new organisms in here that are going to help us understand certainly eukaryotic evolution in a lot more detail in the future. And so with all of this data, another thing that we can do is we can put all of these different organisms together and sort of see who lives with who, or who never lives with who, and sort of describe the social network of the plankton. So we did that using a huge compute, using a random forest approach, and this is the social network in all its ugly glory that we described in one of the papers. So the bottom line from this study is that biotic interactions overall appear to be far more important than abiotic interactions, and that cooperation is far more important than competition. So organisms like to cooperate far more than competing and so on. So our idea of sort of evolution being survival of the fittest and who is the strongest wins is not necessarily valid in this microscopic plankton world. I think that's an important message that came out of this study. And so all of this data is public, right? So everything that we described in these papers are now public. This represents one of the largest contiguous sets of DNA sequence information that is out there in the public domain. These 40 million genes, for example. So this talk is also really a call to come and use this data, because it's what you guys will do with this data that will define the importance of this work far more than what we do with it. Okay, so please go and dig out this data and start using it. The sorts of things you can do with it, besides looking at individual genes and individual organisms, is try to put the biology in the context of the physics and the chemistry of the ocean. So as I said, we tried to collect a lot of physicochemical data during the expedition. And some of this data we brought in from remote sensing, which pulled in from data from satellites, for example, that measure chlorophyll in the surface ocean. Data from Argo buoys, Argo floats, which are out there in the ocean, going up and down to 1,000 meters, collecting data on temperature, salinity, and other things. There's thousands of those out in the ocean measuring as we speak. So we collected all this sort of information that is, again, freely available, publicly available, and made very, I think, detailed contextual data sets for every sampling station that we went through. So this is just one example of Dubrovnik between Croatia and Italy, if you recognize that, where you can see different data that we pull in from satellites and different data that we measure ourselves from the actual sampling on board, data that would go down to 1,000 meters or so in depth. So there's a lot of contextual information there to be looked at, and the sorts of things that you can pull out from this data is, for example, that in terms of the microbial community structure, it's really temperature that is the main driver defining how those structures are organized. So here you see a heat map where we combine we analyzed different environmental parameters and saw which parameters are the most important for defining the results, and we basically saw that temperature can explain on the order of 62% of the variance in the microbial communities. So that's an important result in the context of climate change. As the ocean warms, these microbial communities are likely to change. So that sort of thing can be done using this data. Other things that we have done already, and many other things can be done in the future, is to address how currents disperse plankton. We have, again, very detailed information from physical oceanographers about how water moves around in the ocean, the major circulatory patterns. Here you see a map of the base of South Africa, and you see basically the junction between the Indian Ocean and the South Atlantic Ocean. And you can see how dynamic it is with these rings that are sort of budding off from the Indian Ocean as they go out into the South Atlantic Ocean. So we did a major study of this process, and we found that Southern Ocean plankton are very different from the Indian Ocean, and we found that this region of the ocean circulation is very much a choke point that really limits plankton passage through from the Indian Ocean into the South Atlantic Ocean. So how that bottleneck affects the plankton is very much dependent upon the size of the plankton, depending on whether they're motile, depending on their phylogenetic characters and so on. But that was an interesting result coming out of this. And then we found that these rings, which are known as agulhas rings, bringing Indian Ocean water into the South Atlantic, are very important for remodeling the plankton communities that are in there. So that again was published as part of the special issue of Science. And then another example that we did with the data was to bring together the information about the biology coming from the different sequencing approaches we used, together with the interactome studies, to see how these plankton communities relate to important biogeochemical processes such as carbon export. So this biological carbon pump, which takes organic matter out of the surface of the ocean and takes it down to the bottom of the ocean, we were able to measure that directly on board because we had a sort of macro camera that we put into the ocean, that we put into the water. And we measured, and this took images of the different particles that were in the water column. Okay, so this movie that you see on the top right is basically a movie of the particles in the water column as this camera goes down as deep as 1,000 meters. So from this, we can define carbon export out of the surface, the photic zone of the ocean, and derive carbon export. So that you see on the bottom right, you see a map of sort of carbon export calculated during the whole transect of the Tara Oceans expedition, and you see some regions are pretty active in terms of carbon export, whereas some others certainly are not. So again, we put this sort of information together and did some pretty large computes, and again, just published a couple of weeks ago, some information showing how specific plankton communities correlate with carbon export, communities that are not necessarily expected to correlate with carbon export, including radiolarians, dinoflagellates, and Synechococcus. And we also revealed an unsuspected role for the little guys in the community, the parasites and the viruses. So it's not all about big, fat, juicy particles sinking down to the bottom, coming from large organisms. It appears to be very much parasites, viruses, and so on, which mediate the formation of these large particles, which was quite unsuspected before now. And this is a nice example of a sort of a gene to ecosystem approach that we published a couple of weeks ago. So these are the principal conclusions of the paper so far. The important things that I want to highlight is that all of this data is public, so you're very, very welcome to dig in and start using it. And that the analyses that we've done so far have only been done on 579 samples, so there's 40,000 more to look at. So the data deluge is coming, so better to start now than wait 6 months to dig into this data. And then just some other unpublished things that we're looking at now, just to give you an idea of how we're continuing this work. We're sort of trying to go from single cell up to global sort of processes and back again, using as a basis some of the information we're getting from the microscopic analyses, where in many cases we from this microscopic confocal imaging data we pull out. We reveal a lot of associations between organisms. These are different associations between diatoms and other organisms, which we didn't really appreciate too much before this project. So from this microscopic data, we can now go in and pull out those single cells, those interactions, and study them more at a more global level. One example that I'll show you where we've done that is this curious interaction between a chain of diatoms, which is these sort of banana-shaped organisms at the back, which are forming a chain, sticking onto this ciliate, which is living inside this organic tube that you see inside the interior of the chain. So the ciliate is a bacteriivore. It eats bacteria, not diatoms. The diatom is a photosynthetic organism. Doesn't need the ciliate for anything in particular. So it's a bit bizarre to see this interaction. And looking at the sequence information, and then looking at the global distribution of this interaction, we could then see that go from one sampling station to several other sampling stations and see that the co-occurrence of the 18S sequences of these two guys is quite striking in many regions, in many sampling sites. And going to look at the microscopy data from these different sampling sites, we could pull out different morphotypes of this interaction, where the diatoms is the big guy, or the ciliate is the big guy, and you see in that image above the different sorts of morphologies that we can pull out. And then we can look at the environmental contextual data to see where this interaction tends to occur. And it tends to be principally in nutrient-rich regions. So these guys are not interacting because there's a lack of nutrients in the environment, but they're interacting for some other reason. We're trying to learn more about the interaction by looking at the context of the interaction in terms of the microbiome around the interaction. The transcriptome analysis permits us to look at the gene expression of the two different partners and so on. So that is all coming in the future. And then finally, a major challenge of the project is to get into the eukaryote metatranscriptomes, right? Which is a huge challenge because we have so little information of reference genomes out there. It's very much different to the prokaryotic world, where we have fantastic resources available. For the eukaryotes, it's really a challenge. So this is sort of bar chart of sort of flow chart of how we're dealing with this. We start with total nucleic acids. We extract either DNA or RNA, and then we do metagenomics using the Illumina platforms for everything we're doing so far. So we're sequencing the DNA of different size fractions to derive metagenomes, metabarcodes, and then we're also sequencing the cDNA that we derive from either total RNA or from messenger RNA. And then we try to give this some sort of taxonomic context by bringing in sequences either from reference genomes from strains in culture, many of which have been done by the Moore Foundation, as I'll show you in a second, and also some reference transcriptomes that we've done ourselves, coming from very abundant organisms in our data set. And we're also doing single cell genomics, again, to pull in information about some of the very abundant but totally unknown organisms that we find in our data set. And basically what we have so far, like we did for the prokaryotes, we've been focusing on 68 sampling sites, which you see in that map. And whereas we got 40 million genes from the prokaryotes that were more or less saturating, here we're up to 115 million of the eukaryotes, and we are very far from saturation, as you see up there. So that's a bit scary, but we're more or less working on this data set now. Of this data, about half of them can be given some sort of functional or some sort of taxonomic assignment. The other half, we have no clue what they are. Okay, so there's a lot of work to do there to understand what they are. And so, as I said, the resources from the Moore Foundation are helping us because the Moore Foundation, through the MMETSP program, generated 750 transcriptomes from marine eukaryotes, which in principle should be pretty useful for us, right? So on the bottom there, you see sort of the overlap between the 115 million unigene catalog from Tara and the 15 million or so from the Moore Foundation sequences. And actually the overlap is pretty lousy. Only about 2.5% of our genes overlap with the Moore Foundation sequences. And this, I think, underlines the fact that most of the microeukaryotes that we have in culture are really not representative of the most important organisms that are out there in the ocean. Okay, so there's work to do there to figure out what these organisms are. And there's a plot there that didn't come out, but basically what it shows is that there are about 12 transcriptomes from the Moore Foundation that are the most useful to us in the Tara Oceans data. So these 12 organisms here in the MMETSP data all give more than 50% of their representativity within the Tara Oceans database. Okay, but the other 735 transcriptomes in the Moore Foundation are much less, give us much less coverage. So with that, I will end the grand challenges for the future, and this obviously is a large resource, a large undertaking with many people. Goes way beyond my lab. There's about 23 labs involved, working hard on this project, and we're always looking for new people to join the project. So if you're interested in doing a postdoc on this data, please contact me if you want to come to Paris, or if you want to go to one of the other labs in the consortium. There's plenty of opportunities, and I think I will end there and take any questions. If you don't want to listen to the questions, you can watch this movie. If you don't want to listen to the questions, you can watch the movie instead.
That was really amazing. I was wondering, I just recently learned that there's a lot of relic DNA in soil, that may be over 60%. I was wondering if you know...
mean extracellular
yes,
DNA? Yeah.
Yes. Do you have any sense of how much extracellular DNA is out there in the ocean?
Not really. We did want to specifically sample for extracellular DNA during the project, but we needed too large volumes of water and too many reagents, so it didn't work out. So we don't really know. What we have been looking at is the amount of extracellular DNA that is out there in vesicles, and that seems to be quite interesting. So we look for vesicles in the viral fractions, and we can detect viruses, and we can also detect vesicles, and some of those vesicles have nucleic acid inside them. And the amount of vesicles, the abundance of vesicles seems to depend quite strongly on nutrient content within the water. So there's some interesting things to learn about vesicles and how they may be entrapping DNA and maybe agents of gene transfer or who knows what. So that is going to be very exciting in the future, I'm sure.
So what do you think the prospects would be for, you said that you don't have representative microeukaryote genomes. What are the prospects for reconstructing them from your metagenome data?
I think that would be hard. That would be hard. We've so far only done Illumina sequencing, right? So we have short reads. these genomes can be several gigabases in size, so it'll be a challenge to reconstruct them. It would be great, you know, to use new sequencing technologies, but the timing is not great for us for that because we've sort of burnt our samples already.
Yeah.
In a lot of cases, and for some of these new technologies, you need large amounts of DNA, which we don't necessarily have. So the next expedition, whoever does it, will hopefully do a much better job of that.
All right, great. Well, let's give Chris a hand, and we'll move on to the next speaker.